Okay, my lab studies the molecular abnormalities of the human Rett syndrome brain. The main question that we are trying to address is that what are the molecular properties of the human Rett syndrome brain? We realize that a lot of these studies that have been done in the mouse models of disease have been focused on the male mice, and the male that doesn't have the gene, does not express the gene. However, the patients are usually female patients. These patients do express the gene, however, a truncated form of the protein or a protein that has a mutation on it. So we try to understand the main molecular pathways. So there are specific pathways that we are targeting, and so we look brain region by brain region in a candidate approach and also a genome-wide approach. We have had support from International Rett Syndrome Foundation. We have had support from the Ontario Rett Syndrome Foundation. And part of the results that we have obtained point towards the disease to be affected in different part of the brain more than the other part of the brain. So we have prepared some reproducible systems from the mouse and human cell lines or primary cells, and we genetically manipulate those cells to figure out if you come from the human brain to mouse brain or cell lines, what are the common molecular abnormalities that we do see. We have screened a few FDA-approved drugs that have low side effects. They're cheap, they can be taken orally, and should not cause much of harm to the girls, so they can be tolerated well. And then we are trying those major pathways that we found to be deregulated in Rett syndrome. How these drugs can actually rescue those type of phenotypes in a cell line that does not have MECP2 or have the mutated form. And now we are hoping that we could have some of those data back to the Rett syndrome mouse model, a mouse that has a similar mutation to the human cases, and then try to look at the outcome can be I guess, improve some of the phenotypes. And the patient donations, the post-mortem patient brain donations have come mainly from the Ontario. Some of the other samples that we have received are from NIH Neurobiobank in US. The most recent brain donation that we had was from a 44-year-old male patient that came last year. Our recent results with his brain, analyzing his brain tissue, shows that he does have similar abnormalities as some of the girls that we have examined. However, some part of the brain that previously I guess suggested not to be involved, or some of those cell types seem to be severely affected in the girls. And also the girls are, one of them is 12 years old from Ontario, the other one is 19 years old from Ontario, along with a few other patients that we received from NIH Neurobiobank and the recent male. They all suggest the involvement of certain cellular pathways. One of the things that in a collaboration with a neuropathologist in Manitoba, Dr. Mark Delvigio, we are studying is that how the post-mortem interval would affect these pathways that are regulated by MECP2. For example, if you have a patient that has been died and you're collecting the brain after 5 hours versus 24 hours versus 2 days, how these molecules, detection of these molecules are affected. Because, for example, in case of the last brain donation that we received, the patient was dead for 2 days. We could have the brain collected after 48 hours. So how that interval, post-mortem interval, would affect the detection of these molecules that we look. To our surprise, there are some parts of the brain that are more affected than the other part. This is coming from a collaborative study that we have with Dr. Del Biggio. It seems that DNA methylation itself is not that much affected, but MECP2 per se might be affected in terms of detection in some cells of the brain.